Assalamu alaikum students. So today we'll be continuing with the last topic of our first chapter, and the last topic is two fundamentally different types of cells. So when we began with the topic, I told you that there are two basic types of cells, right? Like cells are of two basic types, prokaryotic cell and eukaryotic cell. So today we'll be studying and comparing the prokaryotic and the eukaryotic cell, right? So beginning from the basics again, that cells are of two basic types, right? Not animal and plant cell, but prokaryotic and eukaryotic cell. Now, what we understood the other day was that pro means before. Karyon means, karyotic is a word that we have derived from the word karyon, that means nucleus, right? So the cells that do not have a nucleus, that do not possess a nucleus, those cells are termed as what? Prokaryotic cells. We also established the fact that all the organisms that belong to this group, they've now become extinct. We do not have any prokaryotes. Apart from what? Bacteria and cyanobacteria. That's called blue-green algae. ये दो examples हैं हमारे पास prokaryotic cells की, right? The group or the kingdom that contains prokaryotic organisms, that kingdom is termed as prokaryote, right? The first kingdom, we call it the prokaryote. ठीक? Apart from it, all other four kingdoms, they are termed, or they are fitted in the eukaryotic cell, right? The eukaryotic cell is the cell that has a nucleus. Now, apart from nucleus, there are other differences as well, and we will be studying those differences today in detail, that what is present in a prokaryotic cell and what is present in a eukaryotic cell. As far as the eukaryotic cell is concerned, we have studied all the organelles in detail. We studied the mitochondria, we studied the Golgi apparatus, we studied ER, we talked about their ribosomes and their functions, we talked about the chloroplast that's present in the case of plant cells, the cell membranes, the cell walls, right? So today we'll be studying a prokaryotic cell in detail. Keep in your mind the cell of bacteria that you studied in the chapter microorganisms and biotechnology in O3, right? We'll be studying the same cell, but in a bit more detail now this time, right? So, the structure of a prokaryotic cell. And then at the end, we'll be doing the differences, right? For now, the structure. What does a prokaryotic cell look like? So, I'm going to divide the cell into two halves, right? This is a prokaryotic cell, a general prokaryotic cell that we'll be studying. I'm dividing it into two halves. One half contains the organelles that are compulsory, that every prokaryotic cell has. And the other half will contain the organelles or the parts that not all prokaryotic cells have, but some have, and the cell can survive without them as well, right? So I'm dividing it into two halves, right? On this half, I'll be writing down the organelles that are a must, the compulsory organelles. Compulsory. Compulsory organ or parts. I'm not calling them organelles because prokaryotic cells do not have organelles, right? And here are the optional ones that may be present in some organelle or organisms, and they might be absent in others depending upon their function, depending upon their mode of life, depending upon how they live, right? So, starting with the compulsory parts first. First of all, every prokaryotic cell has a cell wall, right? The outermost boundary in the case of a prokaryotic cell is a cell wall, with a difference of the composition, right? So the outermost boundary is a cell wall. The difference is the composition. The cell wall in the case of prokaryotic cells is not made up of cellulose, and this cell wall is not made up of chitin either. In the case of fungi, the cell wall is made up of chitin, right? But fungi is a eukaryotic organism. Similarly, in the case of plants, the cell wall is made up of cellulose. These both chemicals do not make up the cell wall. So you can say that the similarity between prokaryotic and eukaryotic cell somehow is the presence of a cell wall. But the composition is entirely different. In the case of the cell wall of a prokaryotic cell, the cell wall is made up of a chemical known as peptidoglycan, or you can call it murine. So this is the chemical, and peptidoglycan, as the name indicates, peptide here represents proteins, glycan here represents the carbohydrates. So this is made up of proteins and carbohydrates somehow, right? But it is not made up of cellulose, it's not made up of chitin, right? The chemical that makes up the cell wall is peptidoglycan. The function is the same. The function is to protect the cell when it is filled with water, right? When it is in turgid conditions, right? So this is what, and to give it a proper shape, right? So this is what the cell wall does here too. It gives it a proper shape and it prevents the cell from bursting when the cell is filled with water, right? So cell wall is a compulsory organelle that is present in each and every prokaryotic organism, in each and every bacteria, whatever the type of the bacteria is, whether it's a free living bacteria, whether it's a parasitic bacteria, it is going to have a cell wall made up of what? Peptidoglycan or murine that is going to be present, right? The second organelle, or the second part that's compulsory, is the cell membrane. We all know that without cell membrane, we do not have a cell, right? The cell membrane is a must, right? Because cell membrane makes up the cell. We studied this when we started this chapter, that we will not have a cell without a cell membrane. Cell membrane is a selectively permeable membrane, and it controls whatever enters and whatever leaves the cell, right? So this is exactly what the cell membrane does here too. It controls whatever enters and whatever leaves the cell. It is selectively permeable. The chemical composition is the same, with slight differences there might be present, right? But two things that are present in a prokaryotic cell and a eukaryotic cell are cell wall and a cell membrane. Now, the next thing, and that the thing that also makes it a bit different, that is the presence of the DNA. Yes, I'm saying DNA, I'm not saying a nucleus. A prokaryotic cell does not have a nucleus. That we have already established, right? Pro means before, carry on the word from nucleus. The cells before the formation of nucleus, or before there was a nucleus, they are termed as prokaryotic cells, right? So this cell does not have a nucleus. Yes, I'm saying it does not have a nucleus. I'm not saying it does not have DNA. It does have DNA. It does have RNA. It does perform reproduction, right? But without the nucleus. So that means that DNA is somehow suspended in this cell, right? So DNA is present. There is a circular DNA, or DNA is present. And also this DNA is single-stranded. We will come to this when we'll study in detail the structure of DNA. But for now, just remember that eukaryotes have DNA that is double-stranded. The DNA has two strands. In the case of prokaryotes, the DNA is what? Single-stranded, and the DNA is present in the cytoplasm without a boundary, without a membrane, without a nucleus. So there is no nucleus protecting this DNA. The DNA is suspended in the cytoplasm just like all other organelles, right? So DNA is single-stranded. Also, Like I told you that the DNA is suspended in the site. That means this entire area, just like eukaryotes, is filled with what? Cytoplasm. So another common factor between eukaryotes and prokaryotes is what? Cytoplasm, right? So they have DNA and they have what? Cytoplasm, right? The sixth common organelle, and that is also a similarity between prokaryotes and eukaryotes, is ribosomes, right? A prokaryotic cell has ribosomes, and these ribosomes perform the same function as the ribosomes in the eukaryotic cell, just their size is different. In the case of the prokaryotic cell, the size of the ribosomes is 70S. They are a bit smaller. In the case of the eukaryotic cell, the size is 80S, right? The ribosomes are 80S in the case of eukaryotic cells, and the size of the ribosomes in the case of the prokaryotic cell is 70S, right? So they're a bit smaller as compared to the ribosomes in the eukaryotic cells, but the function is the same. And I hope you all remember the function. The function of ribosomes was protein synthesis. So it is going to do the same, perform the same function here as well, and that is protein synthesis, right? So these are some of the compulsory organelles. That is, they'll all have a cell wall, they'll all have a cell membrane, right? The chemical composition is different. The cell wall is not made up of cellulose or chitin. It's made up of peptidoglycan or murine, right? There is a cell membrane that is same. It's selectively permeable, controls cell trafficking, allows some molecules to pass through, stops the others, right? Then there is going to be a DNA, but the difference is that DNA molecule is not going to be placed inside the nucleus. There is not a nucleus that is protecting the DNA. The DNA, just like all other chemicals, all other enzymes, all other factors, is suspended in the cytoplasm. Then all the cells of a prokaryotic organism, all of them will have cytoplasm, just like a eukaryotic cell has cytoplasm. The difference is the cytoplasm will be a lot more thicker. Now, think about it. Why will be it a lot more thicker? Why will be it a lot more denser? The reason is simple. In the case of eukaryotic cells, all the enzymes were compartmentalized, right? I hope you remember that respiration was performed by mitochondria. So respiratory enzymes would be in mitochondria. Similarly, photosynthesis was done by chloroplast. So the enzymes responsible for photosynthesis would be in chloroplast. But here is the case is different. Everything is going to take place in the cytoplasm. That means all the enzymes, whatever activities the cell has to perform to survive, all those enzymes will be present where? In the cytoplasm, right? Therefore, cytoplasm is a lot more granular, is a lot more denser in the case of prokaryotic cell as compared to a eukaryotic cell, right? Then there would be, always there would be ribosomes present, right? And then you can sometimes use the word that the DNA is single-stranded, or the DNA is termed as what? Circular DNA, because of the way it is placed, right? Okay. Now, coming to the organelles, or the parts, sorry, again, I keep on saying organelles, the parts that may or may not be present, right? So, some of the bacteria or some of the prokaryotic cells may have infoldings of their cell membranes, right? What are these called? We call them the infoldings. What is the function of these infoldings? The function that these infoldings perform is, writing this again, infoldings of the cell membrane. The function that these perform is, I hope you remember that there are nitrogen fixing bacteria, right? The bacteria that convert the atmospheric nitrogen into nitrates for the usage by plants, right? So there are different types of bacteria. So some of them, those bacteria are nitrogen fixing bacteria. Those nitrogen fixing bacteria have enzymes in these infoldings, right? And then these enzymes help in the nitrogen fixation process, right? So therefore that means that only bacteria that have to fix nitrogen, they need these infoldings. Other bacteria do not need these, right? So therefore these infoldings are present only in bacteria where nitrogen fixation has to take place, right? So these infoldings are optional. They're not present in all the bacteria. They're present only in some bacteria, the ones that have to go for nitrogen fixation. Then there's another organelle that might be present, might not be present. Structure, sorry, not organelle. And that structure is called a slime capsule, or you can just call it a capsule. So in some cases, you can call it slime capsule as well. So in some cases, what happens in the bacteria, there is a covering present outside the cell wall, right? The outermost covering is not the cell wall. There is a covering present outside the cell wall, and that covering is called capsule or slime capsule, right? So this capsule or slime capsule helps to give extra protection to the bacteria. You might have heard doctors tell you to complete an antibiotic course even after the symptoms of the disease are gone. For example, if you have cold fever or cough, right? Doctors tell you कि आपने वो 5 दिन दवाई खानी है. पहले 3 दिन के बाद या पहले दो दिन के बाद probably आपके जो symptoms हैं they disappear, right? The reason you're taking medicine is the reason for the disappearance of symptoms is not that the bacteria has died down. The reason is that this slime capsule covers the bacteria when you start taking medicine, right? Because the conditions are unfavorable for bacteria now, right? And what happens? They become inactive. They're still alive. So you have to keep taking the medicine for complete 5 days for them to die down. So basically, this slime capsule is giving extra protection to bacteria, extra protection, the protection that was given by cell wall, and now extra protection is added by slime capsule. But again, the slime capsule is not present in all the bacteria. It is an optional part. Some bacteria have it, the others do not, right? They're the ones that are very strong, that have developed over years. Those bacteria have a slime capsule for extra protection, right? What else? Then there are certain bacteria that have extra DNA in them. And the extra DNA is called a plasmid, right? Till date, we have no idea what this plasmid does. Even if we remove the plasmid from the bacteria survives. Even if you add a number of plasmids, the bacteria survives. So till date, we have not been able to find out that what is the actual function of these extra DNA, right? So what is this plasmid basically? Extra DNA that the bacteria has, right? But this plasmid has played a huge role in biotechnology. We have been using this plasmid to modify organisms. We've been using this plasmid to enter genes into organisms, right? So this has become our favorite tool to enter genes into organisms like bacteria and other organisms as well. This plasmid can be used because we do not have to destroy the cell to remove this. It's easily removable from the bacteria, right? So we do not know what purpose this plasmid serves, right? But we do know that this plasmid is present in some bacteria and it's missing in the others. Then there is one more organelle, part that is termed as pili, right? There are finger-like projections, and these finger-like projections are called pili. Some bacteria have pili, others do not. Now, what is the function of the pili? The pili basically are responsible for sexual reproduction. Usually, bacteria reproduces by binary fission, right? Also remember, there was this question that I was reading in past papers once, that why we do not call it mitosis? हम बैक्टीरिया की डिवीजन को माइटोसिस क्यों नहीं कहते? मतलब एक सेल से आपके पास 2 cells बन जाते हैं. By definition, that is mitosis, right? But that is not mitosis, because, write it down somewhere for your information, for mitosis, the nucleus has to divide. And remember, bacteria do not have a nucleus. Hence, we cannot use the term mitosis for bacteria. We use the term what? Binary fission. It's just a simple prokaryotic cell dividing into 2. It's not the division of the nucleus first and then the division of the cell. It's just the cell dividing into 2, right? Therefore, this is termed as binary fission. So, normally bacteria reproduce by binary fission, right? A bacterial cell divides into 2 cells, right? But sometimes, in some cases, bacteria are able to do sexual reproduction, right? And pili is the organ, or pili is the part of the cell that is responsible for sexual reproduction. That means pili will only be present in the cells that are going to divide by sexual reproduction. The cells where sexual reproduction does not take place, we will not have pili in those cells, right? So this was a generalized idea of the organelles that might be present, that might not be present, right? Also, yes, there is one more. There are bacteria that are motile, and then there are bacteria that are non-motile, right? What is the term? The term is motile and non-motile. Motile are the bacteria that are able to move, right? That can swim around freely. And then there are ones that are not able to move. They may be parasitic, they move through our blood or our different fluids, right? So if the bacteria is motile, if it is going to move, there is an organelle present that is termed as the flagella, right? So a bacteria might have a flagella. Flagellum. But the position of the flagella, the number of flagella, that can vary. A bacteria can have a tuft of flagella on one side, or it can have a tuft of flagella on both sides. It might have flagella all over its body, or it might have one flagella, or it might have 2 flagella. So their number varies from organism to organism, from species to species. Different species of bacteria have different number of flagella, different positions of flagella. They might not have a flagellum. So this is like not a compulsory organelle, that this is an optional organelle. The organisms that are motile, right? The motile bacteria, they have a flagellum. The non-motile bacteria, they do not have a flagellum, right? So I'll just quickly go through it. I'll quickly review this. We were doing the structure of a prokaryotic cell, right? And we began with the compulsory organelles. The organelles that, parts that every prokaryotic cell has. Primary part is a cell wall, the outermost boundary in most prokaryotic organisms. Then the cell wall is made up of what? Peptidoglycan or murine, unlike the eukaryotic cells. Then there is what? A cell membrane present. Every cell has to have a cell membrane. Then there is a DNA, but the DNA is single strand, also it's circular, right? Then there is cytoplasm and there is ribosomes. The difference is the size of the ribosomes. The function is the same, the placement is the same, but the size is different. They are 70S, whereas in the case of eukaryotic cells, we have 80S ribosomes. Then the optional organelles, beginning with the infoldings, right? Infoldings of the membrane. These are present only in the nitrogen fixing bacteria that have to fix atmospheric nitrogen and convert it to nitrates. Then there is what? A slime capsule present. The outermost boundary is called the slime capsule. In some cases, it is to provide extra protection to the bacterial cell. Then there is an extra DNA, which is known as what? Plasmid. Some bacteria may have it, others may not have it, right? Then there is a structure known as pili. What is the function of pili? Pili have to be responsible for sexual reproduction. The bacteria that are able to reproduce sexually, those bacteria have pili in them, right? Then the last organelle that we talked about was a flagellum. A flagellum may or may not be present depending upon the bacteria's type, whether it's motile or it's non-motile. The bacteria that are able to move on their own freely, they are termed as motile. The ones that are not able to move, they are termed as non-motile, right? So the motile bacteria, they have a flagellum. The number of flagellum and the position of flagellum may vary, right? So I'm quickly writing down the differences, right? There are a few terms that I'll be using that you have to familiarize yourself with, right? So a prokaryotic, and the differences are mentioned in your book. I'm just writing them down here to explain the differences to you, right? On your book, it's on page number 22. You can open that up with me if you want right now. So we have the prokaryotic cell on one side and the eukaryotic cell on the other side. The basic differences, right? So number one is the size. There is a huge difference in size, right? The size of a prokaryotic cell, 0.5 to, it goes from 0.5 to 5 micrometer. They're tiny cells. In the case of the eukaryotic cells, they are 10,000 times greater in volume as compared to a prokaryotic cell, right? Second difference is definitely there is no nucleus. And here they have a nucleus. The other difference is circular DNA. Circular and single-stranded. In the case of a eukaryotic cell, it's not circular, it's linear and double-stranded. DNA, right? The fourth difference that you need to get familiar with, we use a term that it has naked DNA, right? And here, the DNA plus proteins are present. Or you can say that we do not use the term, it's not naked DNA. Now, what do we mean by naked DNA? DNA folds itself to form chromatin thread, right? DNA folds and it gives rise to chromatin. That's how in normal cases it works, right? While the DNA is folding, it fixes proteins in it, right? Imagine your hand free. When you put your hand free in your pocket or in your purse, what happens? You open it up back. It won't open up immediately, right? It'll become entangled, right? Similarly, the DNA is being folded, it's being fixed into a tiny space, the nucleus, right? When it is being folded, what the nature has done is, during those folds are very regular. And in those folds, what happens? Protein molecules are fixed. But this happens only in the case of eukaryotic cell, because in the case of eukaryotic cell, the DNA has to be fixed in the nucleus, right? In the case of the prokaryotic cell, no nucleus. Hence, this is not needed. Now, because the DNA is folded without proteins, hence it is called naked DNA, right? I'm writing it down in bracket. What do we mean by naked DNA? Folded without proteins. Because it is folded without proteins, hence we use the term naked DNA for it, right? And in this case, because it is folded with proteins, hence we use the term that this DNA is not termed as naked DNA. Then we also study about the difference in the cell wall. In the case of cell wall, the cell wall is made up of peptidoglycan or murine. In this case, the cell wall is made up of cellulose or chitin. Plants have the wall of cellulose, whereas fungi have cell wall made up of chitin. Difference number 6 is the ribosomes. I'm writing it down here. The difference number 6, prokaryotic, eukaryotic. Difference number 6 is the size of ribosomes. Ribosomes are 70S, and here the ribosomes are 80S. Another difference, no compartmentalization. Here, compartmentalization. Division of labor is missing in the case of prokaryotic cells. Division of labor is present in the case of eukaryotic cells, right? So go through this. This is given on your book on page number 22. Read this thoroughly, right? Read the structure of the prokaryotic cell. We'll be having one more lecture and I'll be showing you a video tomorrow, a video to review all the organelles that we have studied, and one more lecture because we have to cover viruses as well, right? And then inshallah we'll be wrapping up this chapter, right? I'll see you tomorrow inshallah. Allah Hafiz.